Juan Domínguez-Bendala. I hope I'm pronouncing it correctly.
Yeah, that's
so right. Professor Domínguez-Bendala is Director of Stem Cell Development for Translational Research. He is currently Associate Professor of Surgery at the Diabetes Research Institute, University of Miami, Miller School of Medicine. Before joining the Diabetes Research Institute in Miami, he worked at the Roslin Institute in Scotland under the supervision of one of the creators of Dolly the sheep, the first animal which was cloned. It made a huge story. So Professor Domínguez-Bendala obtained his PhD there and acquired considerable experience in embryonic stem cells and state-of-the-art genetic engineering techniques. Working with other DRI faculty and international collaborators, Professor Bendala is currently involved in several projects revolving around the use of stem cells for transplantation into patients with type 1 diabetes, as well as groundbreaking methods for expansion, regeneration of pancreatic beta cells. Today, he'll talk about advances in pancreatic beta cell regeneration, and I'm looking forward to a very exciting talk. Over to you, John.
Thank you very much, Dr. Tariq. Let me try to see if I can share the screen, and here it is. Can you see my screen now?
Yeah.
Excellent. All right. So I'll start the. Yes, today I will, again, first of all, thank you for the invitation, for the very kind introduction, and certainly for the opportunity to share some of the research that we've been doing over the past few years on beta cell regeneration in the context of type 1 diabetes. And I will start by explaining a bit of what the center does. We are a center of excellence at the University of Miami. That's our building over there. And the entire building is devoted to finding a cure for type 1 diabetes. We are a department. We are not a department, actually. We are a division within the university, but in a way, even though we depend administratively from the university, we also have our own foundation. They raise money across the United States and Europe for research on type 1 diabetes. We were created by a group of parents with children with type 1 diabetes many years ago. And the mission that we have is to find a cure for type 1 diabetes in the fastest, safest, and most efficient way possible. So we have more than 150 people there, mostly scientists and also medical doctors, from surgeons to immunologists, stem cell biologists, such as myself. And the idea is to get, across all these different disciplines to find a cure for type 1 diabetes. We are unique in the States, and I would say the world as well, in that everything that we do has to have a clear translational focus, but we are not simply doing that. There are many good places that do basic science on diabetes, and also there are very outstanding places that do clinical trials for diabetes, but we are perhaps the only place that does everything in a cohesive fashion, from basic science all the way to clinical trials. For instance, we have an entire floor where I work, the 5th floor, where we do all the basic science regarding the potential translational approaches for type 1 diabetes. But we have other units, so if something looks promising in there in my floor and we want to advance that to clinical trials, we go to a preclinical development in the 2nd floor. They will test the cells, for instance, that we have in all small and large animal models, and we have the ability to also conduct clinical trials right in the same building. So again, everything that we do has to have a very clear application for the cure of type 1 diabetes. And in this context, it's not surprising that our favorite organ is the pancreas. And it's actually a very unique organ in that, as you very well know, it has 2 very distinct functions. On the one hand, you have the secretion of digestive juices. That's what 90% of the organ does. That's the exocrine compartment of the pancreas. And it comprises the acinar tissue, which is the cells that make the digestive juices, and the ducts that collect those juices and take them to the duodenum for the digestion of your food. But then you have the endocrine part of the pancreas, which are the islets of Langerhans, chiefly the beta and alpha cells that make insulin and glucagon, respectively. And that's the part that we are interested in, chiefly because that's the part that is, attacked in the case of type 1 diabetes, and you need to replenish that in order to get an effective therapy for these patients. Now, if you look at the pancreas under the microscope, you'll see that the islets, which I have highlighted in there, are embedded in the rest of the pancreas. You can see ductal structures here, for instance, but you have the islets that drain all the hormones directly into the bloodstream, whereas the acinar tissue is draining the digestive juices into the GI tract. So 98% of the pancreas is actually exocrine, and only about 2% is made of islets. When you have type 1 diabetes, there's an autoimmune destruction of the beta cells of the pancreas that make insulin. And of course, there's a genetic predisposition. There's a genetic predisposition for all autoimmune diseases. But then there's an environmental factor. We are learning more and more about what those may be in the case of type 1 diabetes. And there's increasing evidence that there's a viral component to it, that there are viral infections that trigger the autoimmunity in people who have, again, the genetic predisposition to developing autoimmunity. So what happens when you have type 1 diabetes? You'll see here to the left, that's a healthy islet in the exocrine tissue. But in type 1 diabetes, there's an invasion of mononuclear cells from the immune system that attack and destroy all the cells that make insulin. And as a result, these patients need to have insulin for life in order to survive. Now, insulin is great, and it's been one of the major advances of the 20th century because it keeps type 1D patients alive. But unfortunately, it doesn't get even close to mimicking what an islet does. And there are many, many years that the sugar imbalances that come with injecting yourself with insulin may lead to many complications, including blindness, kidney failure, amputations, and so forth. But by far, the most dangerous complication of type 1D is hypoglycemia. That's the fear that parents have every time they go to sleep, that their children may not wake up because their sugar levels may go very low and they're not aware of that. And that's a constant reminder that you have the disease, even if you have it very well controlled. So that's a major complication of type 1 diabetes. And unfortunately, there's not many alternatives to insulin. And one of them is pancreas transplantation, which, as you know, is a major surgery and something that you don't want to do for diabetes. You may do it in the context of other systemic failures. In case you need multi-organ transplantation, you may as well transplant the pancreas, but it's not something that you would do for diabetes owing to the high mortality rate. But then we have another approach which is called eyelid transplantation, and that's something that we have pioneered in my center many years ago. It was actually invented by the director of our center, Dr. Camilo Ricordi, back in the year 1988, in which he developed a method by which you can separate the islets from the pancreas and transplant those islets into people who have type 1 diabetes. So the process entails basically you take the organ, a donor organ, and you inject the organ through the major pancreatic duct with an enzymatic solution, and then you put it in a chamber, which is called the Ricordi chamber, and you shake it. And I'm going to show you a video of that now. And after the shaking takes place, all the pancreas gets dissociated into single cells, not single cells, but rather chunks of cells, including the islets on one side and the exocrine tissue on another. Now you go ahead and centrifuge the resulting product, and the islets are lighter than the acinar tissue, so they remain on top. And that way you can actually retrieve that layer that remains on top and put it into patients. So let me show you the video. That's the whole approach. You get a donor pancreas, you process the islets, you extract them, and then you put them into the liver of the patient. So there are other alternatives to the liver that we're exploring now. The liver, it's much better than the pancreas, but it's not an optimal site. We're exploring now the omentum, which is a fold of the peritoneum, as a much better potential recipient site. And we are also we are bioengineering a site for implantation of islets. But as of now, the standard has been for the past 20 years to transplant the islets in the liver. So there's a video, there's a pancreas inside that machine. That's a chamber that is breaking apart the components of the pancreas. And after the process, you end up with a soup, like you see there. That's a chamber that is transparent. You collect the islets from the upper layer, and then you go to the patient, find the portal vein, put a cannula in the portal vein, and infuse the islets. You will see next that they fall by gravity. Those are the islets falling, and then almost as if it was a miracle, then patients are insulin independent. This is a typical example of a patient who had intensive insulin pump therapy. So that was the state of the art prior to an eyelid transplant. As you can see, despite the presence of a pump of insulin, this patient had the sugar levels all over the place, up and down. And then after the transplant, the sugar levels were normalized. And 1 year after the transplant, the sugar levels remained within the normal range. And this patient didn't have to take insulin anymore. So there are limitations. I'm not going to describe them now, but for sure, one of them, the main one, is the fact that there are more than 3 million people just in the United States. I think that it's about 20 to 25 million people, perhaps even more, worldwide with type 1 diabetes, and we cannot satisfy the demand for islets simply by using donor organs. There's simply not enough of those. So we are looking for an alternative source of islets to make it possible. To take this therapy to all the millions of patients who really need it. Of course, there's another limitation, the fact that we need to provide the patient with immune suppression that's for life. And that's the main reason that we don't do transplants in kids, because immune suppressants bring about many other complications by themselves. And that's something that we don't want to do unless there's a clear need for the transplant. But I'm going to talk about this, which this is my area of expertise, the finding of alternative sources of islets for transplantation. Now, this is a figure from a review that we published a couple of years ago. And I described already the procurement of islets from the pancreas. And there are other alternatives as well. For instance, you can also take the non-islet fraction or the exocrine tissue, which again is 98% of the pancreas. And one approach that has been pursued over the years has been the transdifferentiation or reprogramming of those acinar and ductal cells all the way to islets. It was very promising at the beginning. We ourselves have been involved in this part of this area of research for quite some time, but it didn't, so far it hasn't been as promising in terms of the outcomes as other approaches that we are also exploring. Chiefly this one, which is to find progenitor cells in the non-islet fraction, progenitor cells that we may be able to stimulate so that they proliferate and then give rise to new islets. We were ideally, at the beginning, we were of the idea that we could do this ex vivo. In other words, we could explant or extract the progenitors, grow them up, and then once we have enough of those, differentiate them into islets and put them into patients the same way that we do islets. But now we're actually trying to awaken these progenitors within the body of the patient without the need for transplantation. So we'll get to that point throughout the talk. So we came across the idea of exploring these progenitor cells back in 2014, 2015. And we were not the first ones to explore progenitors in the pancreas. In fact, this is an age-old controversy. There are many people who, like us, and we do believe in them, in the progenitors, because our experiences and our experiments have led us to believe in their existence. But there are many people who claim that they don't do anything if they exist. That basically, if you have any regeneration at all of the pancreas in the adult person, it happens through a replication of pre-existing beta cells, for instance. So that's adult cells that replicate, but there's no progenitors involved in regeneration in adulthood. So it's a very, very controversial topic, and you'll find reviews from one side and the other, and to this day hasn't been resolved. We hope that our recent papers have shed more light on the whole debate, and I hope to convince you that there's a good reason to believe that they exist, these progenitor cells exist, and they have the ability to regenerate the human pancreas, particularly in the endocrine compartment. So at the time, in 2014, 2015, we reasoned that we came to this with an open mind. We didn't actually hypothesize that they were there. We didn't believe in them at the beginning, but we thought that if they're progenitors in the exocrine pancreas, they should be able to respond to BMP7. BMP7 stands for bone morphogenetic protein 7. It's a growth factor. It's circulating in your blood and in mine. It's made by the kidneys, and it grows bone, for instance. It's involved in bone remodeling. And BMP7 is in fact used in the clinic for when you have you have to fuse vertebrae in the context of a spinal surgery. You use that as a glue because it grows bone. So it's a growth factor, right? And it does two things. Of course, it stimulates the BMP signaling pathway, but it also blocks the TGF-beta pathway. And those two things, the stimulation of the BMP pathway and the blocking of the TGF-beta pathways, those are the signature of progenitor cell stimulation in many organ systems across the body. So we reasoned that if progenitor cells are in there, they should respond to BMP7 by proliferating. So lo and behold, we took the exocrine part of the pancreas that we get every time we do an eyelid transplantation. We typically discarded the rest of the pancreas, but I told the people who do that in the fourth floor to save some tissue for me. And we exposed that tissue to BMP7, and we started to see proliferation of structures that resemble eyelids. And in fact, when we removed BMP7, they differentiated into eyelid-like structures that contain insulin-producing cells, glucagon-producing cells, all the hormones that you find in the eyelid. They were actually secreting insulin in response to glucose stimulation. And when we transplanted them in animals, it corrected hyperglycemia as well. So clearly something was growing in there in response to BMP7. And when you withdrew BMP7, they differentiated. So that was quite an striking observation at the time. We published that in Diabetes in 2015. But of course, at the time, we didn't know exactly what was growing in response to BMP7. So we spent the next few years doing lineage tracing studies in vitro to determine which were the cells that responded to BMP7. And that's a very long story. The conclusion was that the cells that responded to BMP7 were progenitor-like. They were not adult ductal or acinar cells. They were not islets, pre-existing islets. These were progenitor-like cells that were characterized by the expression of two markers, PDX1 and ALK3. PDX1 is no surprise. PDX1 is the master architect gene of the pancreas. People who have a mutation in PDX1 who are born with this mutation, they are born without a pancreas. It's a very rare mutation, but it exists. And in mice, when you knock out PDX1, they are born without a pancreas as well. So it's not surprising that the progenitors may express PDX1. It's also expressed in beta cells, but at the beginning, it's a master regulator of the whole pancreatic development. And then ALK3 is activin like kinase 3 receptor. That's the receptor to which BMP7 engages in this context. So it's not surprising either that the cells that respond to BMP7 express a receptor for BMP7. So after we concluded this, we set out to look for these cells in the body, in the pancreas. So where are these cells that respond to BMP7 that express PDX1 and ALK3? And again, this is a very long story, but we found them in only one place, which is the major pancreatic duct tree. I mentioned before, and I'll remind you now that the exocrine part of the pancreas, anything that is on islets, is made of acinar tissue, which is making the digestive juices, and then the ducts. The ducts are the ones that collect all these juices, but historically, they've been also thought to harbor these progenitor cells. And up to this point, nobody had ever been able to pinpoint exactly which were the progenitors in the ducts. And we thought that we had found them finally when we saw these cells that were in the duct that express ALK3 and PDX1 in blue, but they were not expressing carbonic anhydrase 2, which is the green marker in here. And it's a marker that until that point, everybody thought it was a pan-ductal marker, a marker that defined a duct. So in other words, if you're a duct, you have to express carbonic anhydrase 2 because what it does is to make bicarbonate that raises the pH and prevents the self-digestion of the pancreas when the the zymogens and the enzymes are secreted into the lumen. So if you are a duct, you have to express CA2, yet we see cells here in the duct that didn't express the CA2 and they express high levels of ALK3 and PDX1. So we hypothesized that these were the progenitors, but we had to prove it. So what we did was, and Fahad did most of this work, we had to sort these cells out. We had to sort the cells that express ALK3, which is not difficult because ALK3 is a surface marker. We had to find a marker for PDX1 because PDX1 is a nuclear factor. And we had to do a bioinformatics analysis to find a surrogate marker that is in the surface. And we found this one, P2RY1, and we validated that as a surface marker, which means that every cell that expresses PDX1 in the pancreas also expresses P2RY1 in the surface. We were lucky in that respect, so that we sorted these cells PDX1, P12Y1, and ALK3. And we plated them after the sorting. So we get the islets. So every time we do an eyelid transplantation, we take the rest of the pancreas, we dissociate those cells, and then we do the sorting. And not surprisingly, when we plate those cells, they resemble progenitor cells, they resemble stem cells. Unlike the controls, the negative fraction, which grew as a mesenchymal layer, nothing grew from there other than fibroblasts and other mesenchymal cell types. In the BMP7 group, we noticed that these colonies grew much faster. So the mitotic rate was actually much higher, 3 times higher in that group than when you didn't have BMP7. So for as long as we had BMP7 in the medium, those colonies grew, and they expressed PDX1 and NKX 6.1. And that's another key marker of the progenitors. They didn't express insulin, they didn't express glucagon or any other mature marker of the pancreas. They were growing as progenitors. So we published this in 2018 as well in Cell Reports paper. And since then, we've been trying to characterize these cells even in more depth than before. And we've been doing that using new techniques. One of them is the use of human pancreatic slices. I'll get to that in a few minutes, but also by single-cell RNA sequencing and proteomics. Proteomics is something that we have in the we've been planning to do for quite some time, but we haven't got to that point yet. So I'm going to describe now the results of our recent paper that we published in PNAS in May about the single-cell resolution of this progenitor cell niche. So we know that these cells that express ALK3 and PDX1 are progenitors, but we had to see if there was a homogeneous population or not. So we took human donors, the pancreas from human donors, and we dissociated the cells of the pancreas, and then we sorted by ALK3 and depleted all the mesenchymal cell types because we didn't want any contaminants that express ALK3, which is, let's be honest, a very ubiquitous marker across the body. But when you deplete the mesenchymal cells, you are enriching for the ductal cell types. And then we completed the single cell RNA sequencing analysis. And this is what we got. So what we got after we grouped the cells, now every dot that you see there, for those of you who may not be familiar with single cell RNA sequencing analysis, every dot is a single cell. It represents the whole transcriptome of a single cell. So the closer the transcriptome is between any 2 cells, the closer they will be in this graphic. And that's why the software gives you different clusters or groups that represent cells that are transcriptionally similar. So we saw many different cell types that are ductal cell types. We saw centroacinar cells, which are at the interface between the acinar tissue and the ducts. We saw this population over here that were transitioning from ducts to acinar, which was very, very interesting. It was something that had been described before only in the context of neoplastic transformation, but we saw it happening at a high degree, even in healthy patients who had no evidence whatsoever of neoplastic transformation in the pancreas. But more importantly, we saw these 2 groups in here that are progenitor cells that were exactly having a signature, a molecular signature similar to the one that we had described previously in the Cell Reports paper, PDX1 positive and ALK3 positive. So we went ahead actually and we plotted that data. Remember, those are ductal cells. And we plotted that against the map, the single cell map of the whole pancreas. That includes also the endocrine cell types. And to our surprise, we saw that those cells, the green cells that are the progenitors, and you can see them in here, it kind of resembles a little bit the map of Europe. So those progenitor cells are the green ones here that would be France. And then you can see how they're seemingly sprouting from there all the other major lineages of the pancreas. You can see the insulin producing cells that would be Spain, somatostatin positive cells that would be Italy, and then you have Great Britain in here, that's the glucagon producing cells. So it seems that they are at the core, at the root, at the crossroads of every major lineage in the human adult pancreas. And that was very cool for us to actually see. But at the end of the day, these are all bioinformatics predictions. And in my view, they have to be taken with a grain of salt until you do the proper validation in a real, live setting. So that's exactly what we did next. We took the cells from those groups that we had identified as progenitors. We sorted those cells by ALK3 bright expression and P12Y1. Those are the cells that you find in there. And then we transplanted them into immune deficient mice. We transplanted the cells under the kidney capsule. And I want to make clear that The cells that we transplanted were sorted, so they were single cells, and they had no expression whatsoever of insulin or acinar markers or ductal markers. Ductal markers, I stand corrected there. Of course, they were ductal because they are ductal progenitors, but they didn't express any acinar marker or islet marker at the time of transplantation. So once we transplanted them, we saw something quite surprising. This is the kidney here in blue, and this is the cells that we transplanted. Despite the fact that we transplanted them as single cells, they self-organized and they differentiated into every major lineage of the adult pancreas. So you have a tiny islet in there. Those are beta cells. You can see them at higher magnification in here. But you also see the acini, which are the groups of acinar cells that are draining actually into ducts that are there in red and white in another figure. So to us, it was quite surprising, not only to see that there's differentiation happening, but also more importantly, that the differentiation is accompanied by a self-reorganization. It's a self-assembly. We had mini organs in there, mini pancreata in there, which was very, very exciting. Not only that, they were functional because when we challenged the mice with sugar, we got human insulin being secreted into the bloodstream of these mice. And more importantly, we accelerated the process and we made it much more efficient when we gave BMP7 or a BMP7-like molecule to the mice. So we gave it, we injected the mice with a BMP7-like molecule, and we got much more proliferation and differentiation of these cells inside the body, which obviously made us believe in the theory that we had that we may potentially use this approach to reawaken those cells in patients and give rise to new beta cells within the pancreas without the need for transplantation. I'm not going to show that because it hasn't been published yet and it's still very preliminary, but we've been making mice diabetic with a chemical called alloxan that kills all the beta cells. So the sugar levels go very high very quickly. And then we've been giving them a course of a treatment with BMP7-like molecules, and we see that the sugar levels go down. And when we go to the pancreas, they have new islets that are forming versus controls where you don't see any islets because alloxan kills everything. We see lots of islets that are sprouting from the very regions that we had identified as containing the stem cells, the major pancreatic ducts. So hopefully, if I give another talk next year, I will be discussing those data with you in more detail. So that's what we presented in that paper. This is, okay, and I'm gonna go back here. What I'm gonna discuss next is the use of human pancreatic slices, which is a brand new technique that has been developed in the past few years. Let me go back there. There you go. And that's a way that we have to actually get an organotypic culture of the pancreas by sectioning thin slices of the human organ. What you see there is a vibratome. That's an instrument that sections any tissue. In this case, the pancreas is embedded in agarose, low melting point agarose, and that gives the whole chunk of tissue a bit of consistency. And when you look at those under the microscope, it's like having a window into the organ, into the native organ. You can see the islets in there, and you can see the acinar tissue organized in the acini, and you have ducts in there. So it's something that it's very striking. This is not a cell culture. This is a culture of the real pancreas. And you can look at regeneration in real time, not in a cell culture where you have dissociated the cells and then you have epithelial to mesenchymal transition or many other artifacts. And this keeps the nature, the native architecture of the organ. And it's in that sense a very useful tool for us. Now, the problem is that the pancreas is very highly irrigated. It has lots of blood circulation, and therefore a high need for oxygenation. And when you section those slices, obviously you lose that oxygenation. And what we saw is that when we culture those slices in normal conditions, what we call transwells, we immediately start to see within 16 hours and up all the way to 24 hours, the formation of anoxic gradients in the core of the slice. In other words, they are dying because of lack of oxygen. So typically in the hands of any other group in the world, the slices are good for about 48 hours before they start to collapse and die. If we wanted to study regeneration, we needed to keep the slices alive for much longer than that. So we have been testing and playing with the notion of giving more oxygen to the cells in the past. And in fact, we have published several papers on that. And we came across, we came up with the idea of inventing a system where the oxygen, rather than coming from the top and diffusing through the culture medium to the cells that you put at the bottom, the oxygen in this case will come also from the bottom. So what we did was to replace the bottom plastic of a regular Petri dish with a membrane which is made of perfluorocarbon, or PFC. Now, it's a movie I always show. That's a rat that is breathing PFC. It was in a movie in the late 80s by James Cameron, the director of Titanic and Avatar. And in that movie, they were actually using perfluorocarbon to go very deep into the Mariana Trenches. They had to go so deep into the ocean that the pressure was going to be so high that anything other than a liquid inside your lungs would collapse them. So in that movie, they are actually, this is not a special effect. They put the rat inside the PFC and the rat is breathing it. And I don't know if the rat is very happy about it, but the fact is that you can take the rat and it's very happy after doing that. So the thing is that we have made a membrane out of that compound, the perfluorocarbon, and now we have the means to provide additional oxygenation. These are some of the devices that we invented, in which the bottom is a membrane of perfluorocarbon. And we prevent completely the formation of hypoxic gradients when we put them into PFC. This is a paper that we published more than 10 years ago. And we prevent completely the greening here. That's hypoxy probe, which binds to hypoxic regions. That's something that I always like to show, that even if you increase oxygen concentration in the incubator, but keep using a plastic dish, you still get these things, because you still get these gradients that, sure, you'll have a lot of oxygen in the top and the surface of your islet or your progenitor cells, but at the core, you still have hypoxia. But you prevent that altogether by using a system like ours. So we have also shown that this system promotes the maturation of beta cells in an embryonic stem cell differentiation setting. If nothing else, because when it comes to beta cells, they consume much more oxygen than embryonic stem cells do. So just to keep up with their metabolic demands, you need additional oxygen. So by placing the cells in a PFC dish towards the end of maturation, you can get enhanced differentiation. And that's an example here. This is something that we published a while ago. So going back to the slices, and what I'm going to describe now is the paper that we published in June in Nature Communications. What we show here is that when we place the slices, not in a regular transwell, but in a PFC membrane where you have additional oxygen coming from the bottom, we prevent completely the formation of hypoxic and anoxic regions. So that way, just by doing that, we were able to keep the slices alive and functional for longer than 10 days. And in some cases, actually up to 3 weeks versus 2 days, which is what takes for a regular culture system to kill your slices. So I'm not gonna go through this in detail, but one of the things that we noticed is that when we culture the slices in there, we preserve oxidative phosphorylation as the main mechanism of energy production, as opposed to the ones that are in transwells, in controls, where they have to resort to glycolysis. And therefore they have to take a lot of sugar from the medium just in order to keep up with the energy demands, as opposed to a much more efficient energy utilization approach in oxidative phosphorylation. So we did lots of functional studies of the slices at 10 days, and they respond pretty much like you would expect of a native organ, the native pancreas, both the endocrine and the exocrine fraction. One of the things that we noticed in particular, and I want to show you this in here, is that while in regular slices in controls, you lose the expression of PDX1 after 10 days, and PDX1 is fundamental for beta cells to actually be functional, that is very well preserved in the slices that are cultured in PFC. So, of course, we are not interested just in keeping the slices alive for the sake of, breaking a record or anything like that. We wanted to keep them alive for long enough that they can offer us a window into regeneration processes, okay? So, just to test the system, what we did was to get slices from a mouse, a transgenic mouse, in which islets are green, beta cells are green, and everything else is red. As you can see, this is a higher magnification of this mouse. And you can see a duct in there, that's red. Only beta cells in the islets are green. And this is a great system because you can kill all the green by giving naloxone to the slices. So you kill every islet in there. And then the question that we asked was, what happens if you are BMP7 now? You have islets now, what happens if you are BMP7? So this is what happens. You have a slice, it's completely red because we have killed all the islets. You can see that it's red. We have BMP7 for a few days, and when we remove it, we start seeing lots of yellow and then green coming up in there. So that was quite striking for us. And when we look at the higher magnification of these slices, the green is coming out from ducts, from the major pancreatic ducts, which is exactly where we had defined the presence or characterized the presence of pancreatic progenitors. That was quite an eureka moment for us, because one thing is to plate the cells, and then you have all kinds of cells in there and they differentiate. There are many things that you can force a cell to do in vitro, because there are many artifacts associated with in vitro culture. But this is a chunk of the pancreas in which you preserve the architecture of the organ. The endocrine compartment is there, the exocrine compartment, the cell-to-cell interactions, and there you can see how new cells that express insulin are being produced from the ducts. And you can see that in real life, in real time, I'm sorry. You can actually make movies of this, and you can see how it happens in real time. So obviously, we don't have the luxury of having transgenic humans. So if we ever wanted to study this in humans, we needed to replicate this red-green reporter system. And we managed to do that by transducing human pancreatic slices with a set of two vectors, two adenoviral vectors. The first one is a double reporter, which is red and green. And then the second one is the synthetic human insulin promoter. That's a very strong human insulin promoter driving the Cre recombinase. So what happens if you transduce your slices with these two vectors? Well, if your cell is a non-beta cell that doesn't express insulin, that cell is going to be red, because there's a stop codon in here that prevents the expression of green. However, if your cell expresses insulin, this promoter will be activated and Cre recombinase will be activated, and then will be at excision, and that cell will be green. And that's exactly what we see in the transgenic mouse. The beta cells will be green, and the red cells are everything else, right? So the hypothesis that we had is that if new beta cells form from non-beta cells, like a progenitor cell in a duct, there will be a transition from red to green. And you could see the yellow, and you can actually quantify that. And that's exactly what we saw. Here's during BMP7 administration, that's a duct region that we have in the square, and that's red. And then it progressively becomes yellow and green. So we are having a transformation of ductal cells that didn't express insulin at the beginning into insulin expressing cells. And we know these are neogenic because all the pre-existing beta cells in there were actually labeled at day one. This is an example. At day one, you label all the beta cells in green that are transduced, but this happens late when you have BMP7. So in the paper that I just described, we did this exhaustively in lots and lots of human donors, slices from human donors. And we saw that there was regeneration of new beta cells. We could quantify that when you added BMP7. After we published the paper, we had the luxury of having access to slices from type 1 diabetic patients. So these are patients who have very few beta cells, and when one of them passed away in the emergency room, unfortunately. And when we got some of the slices from this donor, we exposed them to BMP7, and we saw formation of new beta cells, which is an absolute first in the field. Now, we did this using another vector that is even more complex and informative than the previous one. In this case, we'll have red if you have a non-beta cell, and if you have a beta cell, it will be blue. Why did we change the green for blue? Simply because we have added another marker in there, which is called GCAMP6, which is a calcium imaging marker. So every time a beta cell makes insulin, secretes insulin, there's a calcium influx, and you can image that. The insulin secretion is associated with calcium imaging, right? So you can image that in green, and that way you can see if your beta cells that are forming are actually functional. They're secreting insulin in response to glucose stimulation. Now in here, you can see the red cells. Obviously, it's not like in the mice where everything was red. In here, we depend on the efficiency of transduction. But you look at a couple of regions in number 2 and 1, and there are cells in there that are turning from red to purple, and eventually they turn blue. And that's the formation of new beta cells in a patient who, a donor who passed away from, with type 1 diabetes. So to us, we actually had to take a step back because we show and think about the bigger implications of this. This is a patient who could have benefited from this therapy if he was already in the clinic, like a patient who died with type 1 diabetes, whom we could have treated if this was already advanced enough. it gives you pause, and it also gives you hope that down the line, we may be able to do this for many people living with type 1 diabetes. Not only that, because of the fact that we can generate new cells, but also because of the fact, I didn't mention this before, we've seen these cells, these progenitor cells, in every donor that we've looked at, regardless of the duration of the disease. So some people thought, well, yeah, perhaps you have progenitors, but they will be lost as the disease progresses. Well, we saw those progenitors both in new onset diabetes patients and in those that have been diabetic for 3 decades or longer. So that was quite striking, and it opened us the door. It opened the door for us to think of therapies in which we will give BMP7 to patients and promote the regeneration of beta cells in the absence of autoimmunity, which is obviously something that we have to deal with as well. So that ends the first part of my presentation. I will briefly give you an outline of what we've been doing on the front of generating stem cells. Generating islet cells from stem cells, which is something that I could give a whole presentation about the advances in the field. As you may know, there are already clinical trials going on using human embryonic stem cells for type 1 diabetes. And the idea is to differentiate these cells all the way to beta cells and then transplant them the same way that we do islets, with the advantage that obviously human embryonic stem cells have the ability to grow in a limited manner. So you can get billions and trillions of them. There's no shortage of those as there is for islets, right? So the field has evolved tremendously over the past decade. And as I say, this is already in clinical trials. The idea is to coax or teach stem cells to become progressively beta cells. And for that, you add the growth factors that you see during normal embryonic development in the right sequence. You have to be extremely careful because the same factor that has an effect in here may have the opposite effect 2 days later. And the same factor may have a completely different effect depending on the dose. So it's been a trial and error process over the many years, but now we have the means to generate beta cells that are relatively similar to beta cells, and we are transplanting them. So I'm going to give you our approach to our contribution to the field, if you would like. And that's a paper that Fahad also was the first author on, and is the making of this approach safer for transplantation. So this is a strategy that we developed to prevent the formation of teratomas. And the reason for that is that even in the best case scenario where you have beta cells differentiating into functional beta cells, at best you get about 40% of beta cells, right? 60% of your preparation is things that you don't know about. We haven't even characterized them in depth. And more worrisome for us is the fact that among those cells that haven't differentiated into beta cells, there are some undifferentiated escapees. So those are cells that remain proliferating after you have completed the differentiation. So if you can imagine going to a patient who has been immune suppressed to receive these cells, and some of these cells may actually keep proliferating after you put them inside the body, that's a problem because they will give rise to a tumor. It's called a teratoma. So that's a challenge, and that's something that has been very well studied in the field. And now there's generally consensus that something will have to be done about that. Because even if the newest protocols are actually quite efficient and they don't give rise to teratomas very often, we're talking about an approach that may be applied to thousands of patients, perhaps even millions if it works. And it takes only 1 in a million developing a teratoma for the whole field to have a setback of 10 years. All the regulatory organisms will be behind our necks, and we will have to stop all this for 10 more years until we make sure it is safe. So there's consensus, as I say, that we want these approaches to be safe. And for that, what we've been trying to do is to generate embryonic stem cells or human pluripotent stem cells, including iPS cells, that are safe for transplantation. And not only that they are safe, that also allow for the removal of any cell that gives rise to a teratoma without killing everything else. Because sure, there are people who are working with suicide genes and kill switches that will kill everything if it gives rise to a teratoma, but well, it will also kill the beta cells that you're putting. So we wanted to preserve those beta cells as well. So what we did was to genetically engineer embryonic stem cells with 2 suicide genes. The 2 suicide genes that we are working with are this one, that's the thymidine kinase or TK. And if you express that gene in a mammalian cell, and you also add ganciclovir to that cell, ganciclovir, which is harmless by itself, will be turned into a toxic product that will kill the cell. So that's one of them. And we place that Swiss gene under the control of the telomerase promoter. Telomerase is active in embryonic stem cells and pluripotent stem cells, but it's, you know, the expression is eliminated when the cells differentiate. So if you have a tumor, it's likely to be expressing telomerase. So only tumor cells will be sensitive to thymidine kinase, as well as obviously undifferentiated embryonic stem cells. Now, the other Swiss gene that we put in there is the nitroreductase. And that will kill the cell in the presence of another prodrug called CV1954. So in this particular case, all the cells will be expressing this, so all the cells will die in the presence of CV1954. However, we have put this cassette flanked by LOXP sites, and then we have put the CRE again under the control of a human insulin promoter. So if you differentiate your cells into insulin-producing cells, those cells will express CRE and will eliminate this cassette over here. So what we have at the end of the day is a situation where only insulin-producing cells, where this gene has been taken out and where telomerase is no longer active, will be resistant to both prodrugs, CV1954 and ganciclovir. If you have any proliferative escapee, any human embryonic stem cell, they will be sensitive to both prodrugs, simply because they express telomerase, so they have TK, and they haven't expressed insulin yet, so therefore they still have the nitroreductase. And if you have differentiation of anything that is not a beta cell, they're still sensitive to CV1954 because those cells are not expressing insulin, therefore they still have this. So we have a double fail-safe approach that will kill anything that is not a beta cell in our cultures. So that's what we present in that paper. And we show that after the genetic modification, the modified clone, which we call lambda H1, is identical in all parameters to the parental cell line, with the exception, obviously, of the expression of the two transgenes that we put in there, right? And one thing that we notice immediately is that the parental cell line obviously doesn't happen. I mean, nothing dies in there when you add either CV1954 or ganciclovir. What you see there in green is proliferation, and if you have any apoptosis, it should be red because that would be caspase-3. So again, the prodrugs don't do anything on regular cells. However, if you have the modified cell line, the lambda H1, in control lines, I mean, that means that you are not adding any CV1954 or ganciclovir, you have your standard proliferation, nothing happens. But when you add either CV1954 or ganciclovir or both, you have 100% cell death, and that's quantified in here. Now, this is probably the most important graphic in the whole paper. Here it shows that if you basically differentiate the cells along beta cells, right? We complete the differentiation, as I mentioned before, in the PFC dishes, just to make sure that we have proper beta cells. So consistent with the field, we get about 25 to 30% of insulin-producing cells at the end of the protocol. And in the parental cell line, the one that doesn't have the modification, that doesn't change whether you add the prodrugs or not. So this is with the prodrugs in orange, nothing happens, nothing changes, right? So you have a small percentage of cells that make insulin. Now, if you go to the parental, to the modified cell line, and you don't add any of the prodrugs, that would be the black columns, still very similar to what you're seeing here. About 30% of the cells are insulin-producing cells. However, when you add the prodrugs, that percentage goes up to 80%. That's very significant. So we are enriching in cells that make insulin at the expense of everything else in there. So that's telling you that we are killing everything that is not a beta cell, or not everything, but a lot of it. We are, for instance, not killing many of the cells that express PDX1, but not insulin, which may be in mature progenitor cells, which is good. But we are doing a significant enrichment in the cells that make insulin. Now, importantly, we wanted to see if that will prevent the formation of tumors, right? So we have an assay where we simply inject human embryo stem cells into immune deficient mice, and they grow into a human tumor, a human teratoma. So what we notice is that these tumors form, and they have the 3 germ layers. They grow in place, and you can see that's very big. In some cases, it got to 10% of the body weight of the mouse. And in those animals, when we treated them with the 2 prodrugs, the tumors disappeared completely within 2 weeks. So as you can see, this is the size of the volume, the volume of the tumor, and it completely disappears. By 3 weeks after the administration of the 2 prodrugs, they completely disappear. And then what you see also here is that you can prevent the formation of tumors in the first place if right around the time of transplantation, you give 1 or the 2 prodrugs. With CV1954, it was perfect. So you give it a course of treatment with CV1954, and then no tumors ever form in those mice. With ganciclovir, it seems that the dose has to be incremented because in most cases, we didn't have anything, but in 2 mice, later than it would have happened in the controls, you had a tumor. So that's a cell that escaped the original treatment with ganciclovir. But the good thing is that immediately after treating the cells again, I mean the mice again with ganciclovir, those tumors completely disappear again. So we believe that this approach not only prevents the formation of tumors and enriches for beta cells, but also in case everything fails and a tumor forms, you also have the means to get rid of it as well by giving oral ganciclovir or CV1954. More importantly, when we transplanted these cells that have been treated with prodrugs either in vitro prior to transplantation or after the transplantation, they are as functional as the regular non-modified cells in terms of insulin production and response to glucose stimulation. So the modification doesn't do anything to the functionality of those cells. So we believe that our approach is very timely now that we are going into clinical trials with these approaches, because again, we don't want one single case of teratoma forming in a patient that may be a problem with the entire field for a long time. What we are doing right now in this project is that the original paper that we published was, we induced the modification simply by electroporation. So we have no means to know where the transgenes have landed, but now we had to integrate those transgenes in what we call a safe harbor of the genome, which is a place of the genome that we know is not going to interfere with anything that is happening in the cell, be it a transcriptional, highly transcriptional region, or anything else that may be disrupted by the integration. So we are using a safe harbor, the ASB1 locus, which has been used in the field to insert genes without disrupting anything else. And we are doing that by CRISPR-Cas9 technology as we speak. So the idea will have to have a cell line, which is GMP compliant, something that has been done in a clean room, and that can be used for transplantation of any product, in cell product into humans. Now, this approach is obviously tailored for diabetes, and that's why we are using the insulin promoter, but perhaps it hasn't gone unnoticed that we can adapt this to whichever cell line of interest you have, right? So by changing the insulin promoter by something that is expressed in the liver or in the eye, you can also differentiate cells and get rid of anything that is the selected cell type. So I'll finish there. That's a picture of the people, most of the people who did some of the work, even though the group is much larger. I'm collaborating also with Ricardo Pastori. He's been my partner at the DRI for many years, and our 2 groups have collaborated in this. This is Fahad prior to his leaving, and this is an elevator at the DRI. And but there are many more people who have contributed, and they are in here. I just want to thank the foundation. Obviously, our foundation has given us the means to do a lot of this research, but also the NIH and the American Diabetes Association. And Fahad actually came with a Fulbright studentship, so I also have to thank them. He could have gone anywhere in the United States. I understand that he was one of the top Fulbrighters in Asia, if not the top in this field, and he could have chosen any other lab, but he chose to come here, and his time here has been very, very productive, and we've got very good papers, and more importantly, we have opened new doors that are now for us to explore, and we have so much to do to take these approaches to fruition and convert them into clinical therapies for patients. So I'll stop there and open the floor for discussion if you have any questions, and I'm here for that. So I hope that everything was relatively clear and the connection was good, something that we have no control over, but if you have any questions, please go ahead. Thank you.
Thank you, John. Wonderful. I'm overwhelmed by the wealth of the data, and in particular, seeing from what I say, the basics to all the way to the applied side. In particular, you finished with this IPS. So let's open for questions.
Sir, so I have a question.
Please go ahead.
Regarding the transplantation of the human embryonic stem cells that you modified as islets, I had a question about the transplant rejection. If you have seen any case where they reject the transplant, because we see a lot of news about, for example, when we talk about kidney transplant or liver transplant in patients, so they reject, and most of the people they die of the reaction that they faced afterwards after the surgery. So, did you see any complication about the transplant rejection when the mice shows that they rejected the transplant and did not survive after that?
Oh, thank you for the question. So when we do eyelid transplantation, and that has been the case for the longest time, eyelids are cells. They're not a solid organ, but they come from someone else. So we need immune suppression. Obviously, these patients need to be taking anti-rejection drugs, which is something I mentioned earlier, is the reason why we don't do eyelid transplantations in children, simply because immune rejection, drugs for immune rejection are very harsh, and we have an immune system for a reason. It's keeping you safe from viruses and from cancer. Immune suppress the whole system, it brings about many other complications. So that's why we only use immune suppression for recipients who really need an eyelid transplant, who have very brittle diabetes and are in danger of, hypoglycemia, severe hypoglycemia, and other cases. However, I haven't spoken about this today, but the idea is, of course, and we have other people working on this in my institute, the idea would be to not to immune suppress the patient, but rather to immune modulate the patient. We have now 2 clinical trials that have been approved by the FDA. In one of them, we're going to give the patients not regular immune suppression drugs, but rather what we call low-dose IL-2. IL-2 or interleukin-2, if you give it in a high dose, what you stimulate is the autoreactive T cells, and that's great when you have cancer. And in fact, it's been talked about using high-dose Th2 for cancer. But for autoimmunity, it does the opposite if you give it in low dose. What it does is to stimulate the T regulatory cells, which are good in the context of autoimmunity because it prevents autoimmunity. So we have seen in other autoimmune conditions that when you give the patients low-dose IL-2, you restore the normal balance of immunity, and we want to test that in diabetes as well. Now, for embryonic stem cells, that's for islets, okay? So for islets, we are using immune suppression right now. We are heading towards immune modulation for stem cells. For the clinical trials that we have going on right now, the cells are not, the body of the recipient is not immune suppressed. What they are doing is to put the cells inside an immune isolation device. So think of it like a bag, a little Ziploc bag, which is porous, so it has a membrane that is made actually of a fabric that Gore-Tex, the manufacturer of sports clothing, they actually have this patent on a membrane that is supposed to prevent the entrance of immune cells, but the pores are small enough that the cells won't go in or out, but the insulin can go out and the sugar can go in. So the first clinical trials have been doing this, have been putting the cells using this immune isolation device. So the idea is that you don't have to immune suppress the recipient. Now, I have to say that the first wave of clinical trials didn't work very well. Not because of the cells are already there, but this immune suppression, immune isolation device is not as good as intended. So what happens is that, sure, the cells of the immune system cannot go inside. However, they do form like fibrotic tissue around, and eventually the tissue inside dies of asphyxiation, so to speak. So they're working with better immune isolation devices as we speak. But in transplants that we did right now in the mice, we use actually the only way that we can keep human cells alive in a mouse, because it's a xenotransplantation setting, is if the mouse is immune incompetent. So these are mice that don't have a functional immune system, and that's why the cells grow in there. And these mice are very sick to begin with. Obviously, they are used as a model because they're useful for us to see whether cells, human cells, engraft. But these mice don't have a functional immune system, and therefore we don't see rejection simply because they cannot reject anything. So trying to summarize the answer to your question, yes, we have to use immune suppression in eyelid transplantation. We want to go away from that and move into immune manipulation or immune modulation, which is a much more efficient way of dealing with immunity without the need to kill the whole immune system. And there's also the alternative of using immune isolation, which is to provide a physical barrier, such as encapsulation or a device like this, that will prevent the attack of the immune cells while allowing for the insulin to get out, the sugar and nutrients to come in. So I hope that answers your question, but it's a long and convoluted answer. So tell me if you need any other clarification.
No, so a follow-up question was because, yes, I do get the point that you use the immunocompromised mice, and in labs, we do use that for transplant purposes. But in humans, we cannot compromise the immune system to that extent that it accepts all the transplant tissues. So that was the follow-up question was that if we use immunocompromising drugs in case of transplanting a human patient, so that will lead to the immunity drop of the patient, and that will ultimately of course lead to tumorigenesis or cancer, etc. So how would we be able to reduce the effects of that?
Yeah, but by moving away from that, we don't want immune suppression. It's bad. You have very rightly pointed out the shortcomings of immune suppression. And there are more, actually. Immune suppressants are actually bad for islets. At the beginning, among the immune suppressants, we had steroids, which actually are diabetogenic. They kill the islets. Now they are not as much, but in the long run, they are bad for islets as well. So yeah, we don't want immune suppression. What we want to do is immune modulation. So rather than bring down the entire immune system, what we want to do is to bring down the arm of the immune system that is autoreactive, the one that is responsible for autoimmunity, but leaving everything else intact and functional. So that's the idea that we want to do. Yeah.
Thank you so much.
Yeah.
So Dr. Afzal?
Okay. Yeah. Thank you so much, Juan. It's a phenomenal work. You are really doing great work. My question is from the first section of your talk where you showed that BMP would induce the progenitor cell. So I was wondering whether it's possible in the patient to targetedly, inject the BMP at the place to induce that, progenitor cell to make, insulin.
That's a really, really good question. And obviously that's something that we've been thinking as well, because when it comes to BMP7, one of the main criticisms that we got from the community when we were sending grants and papers is that, well, this is great, it's growing your beta cells, but it's going everywhere if you give it systemically. So what if you are actually stoking a fire by adding fuel to a tumor, or it can go anywhere else? So those were concerns that we had. One of the things that we did was to actually treat mice that were not diabetic with BMP7. Okay, I'm gonna clarify something. When I say BMP7, I may also refer to BMP7-like molecules, because there's smaller molecules that have the same activity. So we wanted to see, we know that when we have a diabetic mouse and we have BMP7, we have proliferation in the pancreas and formation of new beta cells. But what happens if the mouse is not diabetic? Do we also see that non-specific effect? And the answer is that we didn't. We didn't see proliferation anywhere else, not in the pancreas, not in the liver. It seems that, and this is a growing idea in the field, that these growth factors for progenitor cells work whenever it is needed, in a sense that it is permissive for the formation of new beta cells if there's a stress, if there's an insult, if there's destruction of beta cells, there's an environment there that's pro-inflammatory that is conducive to BMP7 having a function. But if you don't have any of that, if you don't have stress or inflammation, it doesn't do anything. In fact, BMP7 is circulating in your bloodstream as we speak. It's made in the kidneys, and it's quite safe, actually, to the point that it's used in the clinic. It's the only growth factor, other than obviously growth hormone and estrogen and other things like that. It's a growth factor that's in the clinic because it's remarkably safe. there was very recently a clinical trial in which they used a BMP7-like molecule for acute kidney failure after cardiac surgery. So it's a completely different application, but at the end of the day, they were looking at the possibility of BMP7 acting against fibrotic tissue formation in the kidney. So this clinical trial didn't work. That's the short story. However, they got to phase 2 and they treated hundreds of patients in the United States and Canada with oral BMP7-like molecules, and there was no side effects whatsoever. So we are confident that BMP7 will work as expected in people who have type 1 diabetes because they have a pro-inflammatory environment in the pancreas, and also potentially with type 2 diabetes, which is also the case in the pancreas, but it should be safe. However, we don't know for sure until we do all the studies. So we are at present time, we are doing lots of preclinical studies looking at proliferation, tumorigenesis, and all those things. And it will take a while until we complete those studies to show that there's an appropriate safety profile for us to request what we call an IND, an investigational new drug application to the FDA, and go ahead to do clinical trials. But now, if we had needed targeting of BMP7, that's something that we also were thinking about, right? And because it could have been the case that, as you rightly pointed out, it's not wise to give a growth factor systemically. So the injection into the pancreas, it's very, very tricky. You would have to inject retrogradely into the major pancreatic duct, and the pancreas is an organ that you don't want to touch unless you really have to. It's back in the abdominal cavity, difficult to access, and very prone to breaking apart and releasing all those enzymes that will give you pancreatitis. So it's actually quite dangerous to tinker with the pancreas. If you can avoid that, you don't do that. And more so if you have to actually puncture the major pancreatic duct and inject something like BMP7. It's something that we consider, but it's actually quite challenging in mice. Mice have very tiny ducts, obviously, and to do it in mice, it's remarkably difficult. So we didn't get to do that. But we were thinking about the possibility of targeting specific molecules like BMP7 to the islets or to the ducts by using approaches like aptamers or dendrimers. So these are nanomolecules that have been used in clinical trials already, and they have a remarkable specificity for targets. And in the case of dendrimers, which is what we did at the beginning, now that has been actually phased out in favor of aptamers, but dendrimers think of them as nanomolecules that you can decorate with specific target binding domains. For instance, in this particular example, we wanted to target islets, right? And islets express a molecule called GLP-1. Therefore, they express it, no, they receive it because they have the receptor for GLP-1. So we added GLP-1 to the dendrimers, and knowing that islets express the receptor, we injected this through the vein of the mouse to the tail vein, and it got to the islets and it bound to the islets and only to the islets because we could see the presence. So it was really, really cool. And then at that point we were thinking, okay, perhaps we can do the same for the ducts. What if we decorate these dendrimers with BMP7 so that it goes that it was difficult because BMP7 again is a ubiquitous growth factor and there's receptors all over the body. So it was difficult to target, but we were thinking about these possibilities. Aptamers are actually based on nucleic acids, and you can screen trillions of them in vitro before you find one that will bind to your cell of interest. So that's something that we were also thinking about, to find an aptamer to bind to the progenitor cells and deliver the BMP7 only to those. But hopefully that won't be necessary. I mean, just thinking about the complexity of that approach and the regulatory problems that we will have to convince the FDA, not only that BMP7 is safe, but also that targeting is safe, we think that we may be okay proposing the oral or systemic administration of BMP7. It may actually be enough for what we want and saving, while considering all the preclinical and clinical evidence that we have on the safety of similar compounds.
So I have a follow-up question.
Thank you so much. You really satisfied.
Yeah, I was going to ask along the same lines which Afzal has asked. So your data showed, or I think along the talk you mentioned that these progenitor cells are somehow there in the chronically chronic patients with type 1 diabetes, like I think you mentioned something like 3 decades, patients with 3 decades old or type 1 diabetes. So it means despite having the genetic predisposition, so excuse my lack of knowledge in diabetes field. So type 1 diabetes is associated with some genetic mutation, what little I know. So if we treat with BMP or any XYZ factor, we can suppress this genetic predisposition or genetic mutation. Is this what I'm learning?
No. Unfortunately, BMP7 by itself, what we'll do is only, which is not enough. I mean, it's not it's not trivial. It will grow new beta cells. It will induce the proliferation of the progenitors, and it will eventually, when you retrieve it, when you remove it, they will differentiate into new beta cells. And that capacity is there. But as you point out, no, the autoimmunity is there. So this wouldn't work unless we do something at the same time to stop autoimmunity, because then the cells that I induce to form, they will be destroyed again and again. So this is actually resemblant of what happens in what we call the medalists. Those are patients who have been with type 1 diabetes for longer than 50 years. And there was a study done at the Joslin Institute in Harvard, looking at the pancreas of these people, some of whom died in their 90s. And we look at the pancreas of those people who have had diabetes over 50 years, and they're still beta cells, but they're not all beta cells. They're like trying to form all the time. So the body is trying to do this, and it's the immune system that keeps them in check. It's like the whack-a-mole game where, you know, you're popping moles as they pop up. So the immune system is always keeping the new regeneration in place. What we are doing with BMP7 is we are significantly accelerating the process. Let me give you an example. In a mouse, if you kill all the islets by alloxan and somehow you keep the mouse alive, you will see eventual regeneration of those islets, but it will take up to a year, and a mouse lives 2 years. So it's half of the life of the mouse. In humans, if the metabolism and turnover of beta cells is much slower than it is in mice. So I'm going to give you another example. We had a patient who had lots of autoimmune diseases. All autoimmune diseases actually, they rarely come along, because when you have autoimmunity, you have this predisposition, and they usually have more than one autoimmune diseases, right? Well, this person had pretty much all of them, including type 1 diabetes, but he also had lupus, rheumatoid arthritis, many. He was so ill that he eventually ended up receiving a whole bone marrow transplant from his sister, who was a healthy donor. Now, as you know, bone marrow transplantation, it's risky, but if you have cancer or a situation like this, it's life-saving potentially, right? So this person actually, when he got the brand new immune system from his sister, he got cured of all the autoimmune diseases with the exception of diabetes. Why? Because, yeah, there was no longer an autoimmune attack, but the beta cells have been gone for a long time. So if this person lived long enough, 50 more years, perhaps you can see them going back to where they were to a reasonable extent. We think that we can accelerate that with BMP7 very significantly. We think that in just a few weeks of treatment, we can get back to the normal beta cell mass that they had at the beginning based on what we have seen in the mice. In the mouse, in 2 weeks, we have achieved what the mouse takes 1 full year to do. So that's the rate at which we expect to increase the process. But as you point out, the immunity has nothing to do with BMP7. If anything, BMP7 has been shown to have some anti-inflammatory effects, but we don't think it's going to help with autoimmunity. We have to do this in combination with other approaches that will deal and tackle autoimmunity. So as I mentioned before, we are now pursuing 2 clinical trials to immune modulate patients with low dose IL-2. And in one of the examples of low dose IL-2, there's an autoimmune baldness, hair loss, which is called alopecia areata, which is autoimmune. And these patients who received a low dose IL-2, their hair grew back, right? So autoimmunity was gone in those patients. So that's what we tried to do with diabetes. We had to stop autoimmunity for my approach to work. Otherwise, the BMP7 will be, we will get new beta cells, but they will be destroyed again. And who knows if there are more progenitor cells after that. So we have to be careful and not to induce the formation of new beta cells before we can deal with autoimmunity.
Thank you.
Other questions? Dr. Faisal has a question. Dr. Faisal, you can ask.
Okay. Thank you, Professor Juan, for treating us to an excellent talk. So, I have a question for the later part of your talk, but just, talking about the BMP7, I think this is wonderful that, you can, you have identified these progenitor cells and you can actually differentiate them to produce insulin. So, as Dr. Afzal was saying, the dangers of the, treating with BMP7, and you very explicitly explained that, there might not be that much risk of treating patients. But I was just wondering whether it's actually necessary to treat people for a longer period of time. I mean, once you have the differentiation, do you still need to keep treating them with BMP7? I think once you differentiate the cells, there must be a timeline for that, and then you don't need to give them BMP7. Is that right?
Yeah. We don't envision this as a chronic treatment by any means. We want to bring back the beta cell mass to where it should be so that it's functional. And then that should be it. I mean, the cells that have been differentiated, they are there to stay unless they, if they die again, we may have to repeat the treatment. But as I mentioned before, who knows if you still have progenitors after that? We don't know if this is a one-shot kind of situation. That's something that remains to be determined. For all we know, the progenitors, we may have a pool of progenitors, and when they become new beta cells, that's it. I don't think so. I am of the opinion that the progenitors will remain in there. There's a niche that will remain in there. And my opinion is based on what I also indicated before about this long-standing diabetic patients who have been diabetic for decades, and you still have new beta cell formations. So that tells me that in spite of the immune attack, you still have progenitors that are trying to do the job. So yeah, I think that BMP7 should be given only for as long as it's necessary until you get new beta cells, and then you have to take care of them, which is another discussion, because when it comes, for instance, to type 2 diabetes, which is associated with obesity and sedentary lifestyle and old age, in those patients, oftentimes the problem with many of the therapies that we are proposing is compliance, right? So what if we have a severe type 2 diabetic patient in which you already have beta cell loss, and we replenish those beta cells, but the patient doesn't do anything about his or her lifestyle? Well, they will be destroyed again, right? So if they were, we hope that BMP7 may help again, but we don't know if this is, we don't envision this to be a chronic treatment. Yeah.
Okay, so my second question is about, these human embryonic stem cells that you treat with these 2 drugs to kill anything other than the beta cells, right? So one of these is a DNA alkylating agent. So I was wondering whether the treatment is actually in vitro, so you treat them first and then you transplant, or you transplant and then treat the animal with those. In either case, even if it kills the non-beta cells, all the proliferating cells, they might actually damage the DNA, and actually that ultimately can lead to cancer. Although you've shown that, till 18 weeks, there were no tumor in the mice, but, it could be different in humans.
Well, you raise a very valid point, and let's be frank, there's no 100% safe approach when it comes to cell therapy. So we have to balance the risks and make an educated decision, right? When it comes to genetically engineered cells, it's all, the jury is still out. We don't know exactly what's going to happen. We can have a good idea based on what happens in animals, but even GFP, something as routinely used in labs across the world, GFP has also been shown to be toxic to animals who have GFP as a transgene. So for sure, there's no way to know if down the line those cells may suffer from it. Now, in the studies that we did in mice, within obviously the short-term follow-up of those studies, which is not so short-term for mice, because again, mice live for 2 years, and we did this follow-up for several months, we didn't see any deleterious effects. And in fact, the function of those cells that we had treated was exactly similar to the one that we saw in animals treated with the cells that had not been modified or treated the animal with the prodrugs. So, so far the evidence is consistent with the fact that we don't have any such long-term harmful effects. However, should we have them, And you pointed out the example of cancer. Let's say that one of these cells eventually becomes tumorigenic. Well, the good thing is that we have already the medicine with the disease, right? So we have already these 2 suicide genes that will take care of any cell that becomes tumorigenic. We can kill it at any time by giving anciclovir or CD1954 to the patient. So we think that we have that covered. Everything will be killed if it misbehaves, so to speak. That's what we expect. But obviously, before we go to humans, there's a lot to be done in mice and looking at the long-term effects of these approaches. One of the things, for instance, that we are most worried about, even more so than the long-term effects of these transgenes on the cell, is the fact that we've been inserting these genes in places of the genome that we have no control about so far. And we don't know what may happen if you are inserting these transgenes in a region of the genome that shouldn't be silenced. So you can silence a tumor suppressor gene, which is not good, or you can activate, an oncogene. So these things have to be carefully considered, and that's why we are doing CRISPR-Cas9 to insert the gene in a region of the genome that is supposed to be safe and not interfering with any neighboring genes. So what you presented is a legitimate concern and something that we obviously have to look at carefully before moving ahead with clinical trials. What I'm going to tell you, though, is that at some point you had to take calculated risk, and even the FDA, I didn't think that they would approve treatments with human embryonic stem cells as soon as they actually did. We have been concerned about teratomas for the longest time, yet they went ahead and approved these treatments in 2007. Way before, not 2007, no, sorry, 2017, way before we had everything, all the answers for it. So, sometimes as I pointed out, there's no 100% safe approach, and you have to balance and carefully see the risk-benefit ratio of any intervention and go ahead with it and hope for the best sometimes.
And also, thank you for supervising such a wonderful student. He seems to have produced an excellent PhD. Fahad, I don't know him, but, it seems like he did great work.
He is outstanding. I mean, he not only a very clever and, fantastic student, but also a delight to work with and a very good friend. And I'm thankful to him for coming to my lab rather than the other way around, I would say. But it's been, if you guys there in Pakistan, if you have students like half of what Fahad brought to me, I want to bring any of them to my lab as soon as possible. So really, really good experience. I really enjoyed my time working with him and look forward to keep collaborating with him now that he's going to become an independent investigator.
So, John, one last question I have is the beautiful data about single cell transcriptomic data you showed for these pancreatic cells, which you showed like a European map, where these progenitor cells somehow seem to be the founding members of all those cell lineages. Did you analyze these different cell lineages and try to deduct some sort of, cell fate code for the pancreatic cell and different cell types?
I'm not sure I get your question. If we detected what?
So if the progenitor cells are somehow the founding cells of all these different cell types, which you showed, like Italian or what different cell lineages which develop from these progenitor cells, did you try to, find some transcriptional code out of, because you have beautiful data which is clustering in different cells?
Right. So no, we didn't study the differentiation, the factors that may be involved in differentiation. It's interesting because we believe that the pancreas, there's a lot of flexibility, developmental flexibility in the pancreas. That's actually very well studied. And that goes even higher when you have stress, inflammation, and all those things, like cells, they differentiate. What we have found, and it is consistent with what others are finding in this field of single cell transcriptomics, is that cell fates are very rarely cast in stone, like inflexible, and that's a beta cell, and that's what you are, and you're going to be forever. There's a gradient. When you look at the transcriptome of every single cell, you can see that there's a blurriness between different lineages, and they bleed into each other. And that's consistent with what we think is happening in the pancreas. We know already that when you have type 2 diabetes and you have a lot of inflammation, and those things are bad for the beta cell, what the beta cell does to protect itself is it dedifferentiates. It goes back to a state of progenitor-like characteristics, stops making insulin, and when the situation is more favorable, it redifferentiates again. So we think that's happening all the time in the pancreas based on a dynamic microenvironment that may be dictated by either any disease status of the patient or a lifestyle or diet. All these things are happening all the time. We detected this population that is continuously transitioning between acinar and ductal cells. So that has been, I think, the major theme over the past, I wouldn't say even 5 years. That's been really, really new. The discovery that lineages are not set in stone. They are actually in a state of flux, and there's a gradient between all these things. And find those progenitors, right? So they are progenitors that are not, you cannot say based on their molecular signature if they're going to become one thing or another. And then you put them into an immune deficient mouse and they become everything. It tells you that they have that ability, and what factors determine whether they go in one direction or another, that's not known to us or to anybody at that time. It will be dictated by the milieu in the microenvironment, and that's something that we have no control over right now, but it's certainly, there's a lot of study in that direction, and we'll be busy for years to come doing that.
Thank you. Thank you. Other question? Are there any questions, Abdullah, on Facebook or no? So let's all thank John. Thank you very much for being so generous with your time. It was a real treat to listen to your work. And I think like what Faisal said, I'm really happy to see Fahad also doing so great, and you mentoring him all the way like a wonderful mentor. Thank you very much. I think we will bother you more than often because your area is such an exciting area. Despite being such a big time zone difference, I think even at 12 o'clock midnight, you will see these people sitting here excitingly or anxiously looking forward to listen to you. Thank you very much.
Thank you. Thank you so much. It's been my pleasure, and anytime. Okay.
Thank you. Have a good day there.
You too. Take care.
Have a good day, sir.
Bye-bye.